Namaste everyone. My name is Gargi and Shabaray. I am a botany teacher. So today I have an amazing topic to discuss with you all, that is about androecium. We are basically speaking about sexual reproduction in flowering plants. Flowering plants, when I say, it means angiosperms. Today we are specifically speaking about androecium, the structure of anther, and then we will be studying how anthers will develop, will produce pollen grains, okay, with the process called microsporogenesis and microgametogenesis. Okay. So, androecium, andro is something related to male. Okay. So, androecium is the male reproductive part of the flower, and if you see a flower, what are the four whorls present in it? Outermost is calyx, then inner to that is corolla, third one is androecium, third inner to corolla is androecium, and innermost is gynoecium. So, when I draw it, how does it look? Just giving you a gist. So, this is calyx, then there is beautiful corolla, right? Then there is androecium, and then there is gynoecium. So, all of these are located in the thalamus or the receptacle. They are placed on the receptacle. So, this is a typical flower which has got calyx, corolla, androecium and gynoecium. Of course, their individual structures are different. I mean, they have lot of structures included in them. Okay. But today our topic is going to be androecium. So, what are the individual units of androecium called? They are called as stamens. Okay. So, stamens, if you take one single stamen, it looks like this. I will use yellow color only so that I can refer to this. Yeah. So, this is an individual stamen. Okay. So, stamen has got three structures in it. One is filament, which helps it to attach to the flower, and then above there is anther, which actually produces pollen grains, and in between there is a structure called as connective. Okay. So, this connective, as the word suggests, it is the one which connects the lobes of anther. Okay. So, this is the structure of stamen. So, there are three structures present in stamen. What are they? Anther, one second. Yeah, anther, connective and filament. Okay. So, now if I have to look into the anther, I will go to the next slide. So, this is a diagram which I have already kept ready, but before that, before going to that, I would like to draw the structure of anther in specific. So, this is an anther. Of course, this is the filament. I am not going to emphasize on filament now. My topic is anther. Okay. So, each anther is said to be bilobed. Bilobed because it has got two lobes in it. Okay. And in each lobe, it has got two curves, or I can say they are called as theca. Theca means they are, ridges like or bulged structures. So, altogether it has got four chambers. Okay. So, if I have to take a cross section of this, how does it look? So, it will look like this. with this diagram you will understand the structure of anther more clearly. Like this. So, okay, so now I will draw it like this. Now you see there are two lobes in it. One second. Yes. So, when I say anther is bilobed, very important. Bilobed means it has got two lobes. So, one lobe is say this one, or I can say like this. I'll consider it as I have kept it like this. Okay. So, this is one lobe and I'll use different color, and this is another lobe. So, you can imagine they are kept like this, like this. So, this is one lobe and this is another lobe, okay. And in each lobe, if you see, it has got two theca. See, this is one theca and this is another theca. This is one theca and this is another theca. So, they are bilobed and they are dithecous. Dithecous. So, bilobed, two lobes, and in each lobe there are two theca. So, it is bilobed and dithecous. Now, all together it can incorporate 4 sporangium. Okay, sporangia is plural, sporangium is singular, and because it is male reproductive structure, we call it as microsporangium or microsporangia. Okay, so here each anther can incorporate 4 microsporangia. Okay, that's why we call it as tetrasporangiate. Tetrasporangiate. So, bilobed, dithecous and tetrasporangiate anther. Okay. So, this is the overall morphology of the anther, and if I take a cross section of that, how does it look? It looks like a butterfly actually, and what is its anatomy we will learn now. I hope this is clear. Okay. So, when I take the cross section of an anther, it has got 2 layers. Basically, it has got 2 layers. I will use a different color. So, one is parietal layer, and second one is sporogenous layer. Okay, so it is made up of 2 layers. One is parietal layer and the other one is sporogenous layer. What is this parietal layer? Parietal layer is something which is present on the parietal side, and it is meant basically for protection. Okay, whereas sporogenous, the word itself says spore genus, genus is generating. So, spore generating layer, that is sporogenous layer. Okay. Now, this parietal layer is divided into 4 layers again. Okay. So, if you see the anther, outermost single layer is made up of epidermis. You might have studied anatomy of plants. All the plant structures, it is the outermost layer is epidermis. Okay, and epidermis is of course meant for the overall protection. Next to that is endothecium. Endothecium is also a single layer, but it has got slightly larger cells. Okay, and usually even if epidermis ruptures or other layers, other inner layers rupture also, this endothecium will not, until and unless the anther is totally matured. Okay, only once it matures, it will help in the anthers dehiscence and pollen's dispersal. Okay, so that is endothecium. Endothecium is meant for protection, which prevents the rupturing of the anther in immature, I can say it prevents immature rupturing of anther. Okay. Then next to that is middle layers. I am not telling middle layer, but middle layers. So what does that mean? It is not just one layer. It is made up of 2 to 3 layers, which is meant basically for nourishment of the pollen grains. There are developing pollen grains in these spots, right? So they are giving nourishment for those developing pollen grains. And innermost, the most important one is tapetum. See, these brown colored ones are middle layers, yellow one is endothecium, white color is the epidermis. Okay. Now you can see the blue layer, it is the most important and innermost layer among the parietal layers. Okay. So what does this mean? Tapetum is usually multinucleate. Tapetum is multinucleate and it is dense with cytoplasm. What is the need of having so many nuclei in each cell, and what is the need of having dense cytoplasm? Because it is like, you know, mother layer in the sense nursing layer for the developing sporogenous tissues or pollen mother cells, whatever the word is, we will be learning about them next. Okay. For the development of microspores, tapetum plays a very, very important role. It is multinucleated and dense with cytoplasm so that it can give enough nourishment to the growing microspores. Okay. So that is about the structure of anther. So anther is bilobed. Bilobed means it has got 2 lobes, and each lobe, if I hold it like this, how does it look? Yeah. So one lobe is present here, I mean one thecus is present here, another theca is present here. So it is bilobed and each lobe has got 2 theca, so we call it as dithecus, and in total each anther can incorporate 4 microsporangia in it, so we call it as tetrasporangiate anther, bilobed, dithecus and tetrasporangiate anther. Most of the 99% of the plants have bilobed dithecus anther only, but in case of this one hibiscus, it has got monothecus anther. Okay, monothecus anther. So it is not tetrasporangiate, it is just bisporangiate. Okay, and the if you take the cross section of anther, it has got 2 layers, parietal layer and sporogenous layer. Parietal layer is again made up of 4 layers, that is epidermis, endothecium, middle layers and tapetum. Epidermis is for protection, okay, and endothecium is for prevention of immature rupturing of anther. Immature, I will write rupture of anther. Okay, and middle layer is for nourishment, and lastly tapetum, that is the most important layer, which is multinucleate and dense with cytoplasm, and it is very, very important to provide nourishment to the anther. Here both tapetum and middle layers are meant for nourishment only, because in the course of time middle layers are all going to degenerate, because microspores will grow in size, anther also will have to grow in size, microspores also will grow in size, and that time the this pollen sacs, I forgot to mention the word for this, these are called pollen sacs. Sac is a portly or pouch like structure. So there are 4 pollen sacs, and here pollen grains are going to develop. Okay. So here pollen grains are going to develop. So during the course of time, these middle layers and all are going to get ruptured or degenerated, I can say. So ultimately tapetum is the one which primarily helps in nourishment for the developing pollen grains. I hope this concept is clear to you all. Okay, so that is about structure of anther. Proceeding to the next slide is about microsporogenesis. So when you have heard the word genesis many times. So here I use the word microsporogenesis. Microspore is the spore which is produced by the male reproductive part of a flower. So we call it microspore. If the spore is produced by female reproductive part of the flower, we call it megaspore. Okay. So now we are speaking about the male reproductive part, and obviously we will have to see the study of the production of microspore. So we have the definition of microsporogenesis. The process in which the sporogenous tissue undergoes meiotic division to form microspores within the anther of a plant. Okay. So there is a structure, there is a tissue called as sporogenous tissue. I had told earlier, sporogenous layer. So that is the layer which produces sporogenous tissue. What is a tissue? Group of cells. So they are all sporogenous cells. What is sporogenous cell? Spore generating cells or microspore generating cells. So we call it as sporogenous tissue. It undergoes meiotic division. Why does it have to undergo meiotic division? Because its ultimate agenda is to produce gametes. So the plant is diploid. So can its gametes be diploid? No, right? Gametes always have to be haploid in order to, because after fertilization again they have to restore the diploidy, right? So sporogenous tissue undergoes meiotic division. This is very, very important. Undergoes meiotic division to form microspores within the anther of a plant. Okay. And next is after microsporogenesis. What is the result of microsporogenesis? It is microspores. We are getting microspores from microsporogenesis. What is the fate of microspores? Microspores will have to develop in size, and ultimately they will produce 2-3 celled male gametophyte. What is a male gametophyte? It is nothing but pollen grain in case of plants, and this pollen grain will be containing male gametes. So microgametogenesis is the formation of male gametes from microspores. Okay. So how is this going to happen? We will go through now. I am going to write a flow chart so that you understand it better. Okay. Now during when the flower is just formed, it is not bloomed yet. That time if you see the anthers or the stamens, they will be still young, right? So initially what happens, there will be certain cells, basically there will be only one cell, that is archisporial cell. You might have heard about archaea or archaebacteria. What do we mean by archae then? Archaebacteria means ancient bacteria. So archisporial cell means it is the initial cell or ancient cell. So there is one single archisporial cell. Okay, archisporial cell, only one will be there. Okay, and you have to remember its ploidy, children. It is diploid. Okay, now this archisporial cell, it will undergo periclinal division. Okay. So see, there is one cell. Okay. If a cell undergoes division like this, means if the division is happening like this, then you call it periclinal division. Periclinal division. Okay. Okay. If there is one cell, and if the division is happening like this, such that two cells are formed like this, then you call it anticlinal division. So what is the division I mentioned here? Archisporial tissue is, archisporial cell. Okay, there might be many cells also, not many in the sense, very few. Okay, maybe 1 to 5, that range. It is in single digit, this thing. So archisporial cell that undergoes periclinal division. So how have they formed? It is one cell above, or one cell on the outer side and one cell on the inner side. So if this is the structure, there was one cell, and now the same cell has undergone division like this. So it has undergone division like this. Okay. So this outer cell is known as, outer cell is known as parietal cell. Why is it called parietal cell? Because it is going to form parietal layer. Okay, and inner cell is known as sporogenous cell, because it is going to continue dividing through mitosis to form sporogenous layer. Remember children, in these cases the division is mitosis, meiosis has not occurred yet. So archisporial cell is undergoing periclinal division, wherein two cells are formed, one on the outer side and the other one on the inner side. You can see here outer cell and inner cell. So outer cell is the parietal cell because it is going to give rise to parietal layer, and this one inner cell is called sporogenous cell because it is going to give rise to sporogenous layer, or sporogenous layer. Okay. Now parietal layer we have already discussed. What are the cell layers? Epidermis, epidermis was also there initially, endothelium, middle layers, all these structures will be formed. Okay. Now let us talk about, yeah, that's okay. I am going to write it again in a better color probably. Yeah. So archisporial cell which was diploid, it has undergone periclinal division. Outer parietal and inner sporogenous layer. I am not going to speak about parietal layer. Let me directly go to sporogenous cells. Sporogenous cells continuously divide through mitosis and it will form sporogenous tissue. Sporogenous tissue is also diploid. Okay. Now among these sporogenous tissue, 90 to 95% of sporogenous cells that will grow in size and they will form pollen mother cells, or they are also called as microspore mother cells. So sporogenous cells grow in size because they have lot of work to do. They should have enough energy, right? So they will form pollen mother cells, which are abbreviated as PMC, or they have another name, that is microspore mother cells. They are haploid or diploid? They have just grown in size. So definitely they are diploid. Next, what happens? These pollen mother cells, they will undergo meiosis. How do I represent meiosis when I write flow chart? like this. This is how I personally write meiosis, represent meiosis. Okay, so they will undergo meiosis. Meiosis to form what? What is, see, in meiosis what happens? One diploid parent cell will produce how many? 1, 2, 3 and 4 haploid daughter cells, isn't it? In meiosis, one parent, because you have to have the strong fundamentals regarding meiosis and mitosis differences, that's why I am just mentioning. In meiosis, a diploid parent cell, it will give rise to 4 haploid daughter cells, whereas that's why we call it as reductional division, because the ploid is reduced, diploid has become haploid, whereas in mitosis, if it is a 2n, then it will form 2 daughter cells which are 2n only, or if it is n, daughter cells also will be n only. So it is called equational division. Just for your understanding, I am going to rub this again. So mitosis is reductional division and meiosis is reductional division and mitosis is equational division. Okay, so now these pollen mother cells or microspore mother cells, they undergo meiosis to form microspores. But these microspores, see, from each pollen mother cell, they will form 4 microspores, right? And it is a product of one single microspore mother cell. So what happens is, all those 4 will be together as a cluster. Those 4 microspores will be together as a cluster, and that's why we call it as microspore tetrad. Okay, so pollen mother cell, it will produce microspore tetrad. Often a lot of neat questions will be asked on this concept. Suppose, see, one pollen mother cell is going to produce 4 microspores. One PMC is going to produce 4 microspores. Okay, then how many will 50 PMCs produce? If there are 50 pollen mother cells, how many microspore mother cells will be produced? So you will have to multiply it with 4, that's it. It will be 200 microspores. So these kind of questions are very commonly asked. So you need to understand this concept, children. So archisporial cell, which was diploid, it produced sporogenous tissue which is diploid. They grew in size and formed pollen mother cells which is also diploid, and they underwent meiosis to form microspore tetrad, which is diploid or haploid. They are haploid, and as I said, how do they look like? As I said, they are produced in a cluster like this. Like this. Or you can imagine a ball which is formed of 4 microspores. Okay, so it looks like this, and they are all attached together. But in the course of time, as anthers mature, more and more water will be taken up by these microspores, and there will be dehydration happening inside the anther, and here they are attached together because of that water content, right? But as the anther matures, what happens? They dissociate, they get separated, and they will separate, or you can say segregate or dissociate, whatever word, and they will form microspore ultimately, which is N. Okay, so this is a microspore tetrad which are held together as a single unit because they are produced from one single pollen mother cell. Then after the anther matures, because of dehydration, that gelling thing will be lost between them, and they will separate, thus forming microspores. Okay, and there occurs questions like say there are 400 microspores. There are 400 microspores in a pollen sac. So how many pollen mother cells have produced them? So for one pollen mother cell it produces 4. If there are 400 microspores, I will have to divide it by 4 and I will get the answer as 100 PMCs. Okay, so this is how microsporogenesis happens. I hope this is clear to you all. Okay, so next we will proceed with what is the fate of microspore. Okay, of course this microspore, it will develop two walls, intine and exine. Exine is made up of the most resistant material called sporopollenin. I think I won't deal with that currently, but what happens internally we will discuss. Of course pollen grain structure and all we will discuss, but in the next concept. First we will have to understand how pollen grain is formed and what is microspore, what is pollen grain and what is male gametophyte. So yes, now I will draw this microspore again. Okay, this is the microspore. Now what happens, individual microspore, it will grow in size. Okay, so as it grows in size, a lot of cytoplasm, reserve food materials and all will be filled in it, and once we, when do we say that it has grown in size or when it has become matured? That is when it develops a vacuole. Okay, so microspore will grow in size. It will be filled with lot of reserve food material and lot of cytoplasmic components. Of course, nucleus is also there. Then I'll draw the nucleus in yellow color so that it is easier for us to understand. Okay. Next, what happens? Microspore, it will start developing vacuoles in it. Okay. So this is still a microspore. What is the ploidy of microspore? It is haploid. Remember, no more meiosis happens again. Okay. So in this microspore, next what happens is this one nucleus, microspore nucleus is there, no? That will undergo mitotic division to form two cells. Okay, it will undergo mitotic division to form two cells, and these two cells are like unequally divided, in the sense one cell is larger and the other cell is smaller. Okay, now the nucleus, this thing, it has got microspore nucleus, right? Now what will happen, this nucleus will undergo equal division, like it will undergo mitosis, the nuclear division also will happen. Okay, but the cells which are formed, they are of unequal size. Nucleus is equally divided, equal division, equal proportion of materials are there, but the cells which are formed as a result, no, they are unequally divided. So how do I draw this? Okay, so this is the larger cell and this is the smaller cell. Okay, so nuclear division has happened equally only, but the cells are formed like unequally, means one is larger cell which is called as vegetative cell, vegetative cell, or it is also called as tube cell. Okay, and the other one is known as generative cell. Okay, why is it called a tube cell? Because it is ultimately going to give rise to pollen tube. Okay, and this nucleus is called vegetative nucleus. Vegetative nucleus, and this is known as generative nucleus. Okay, first of all, I'll come back to the concept. This is microspore. Microspore has grown in size, it has developed vacuoles in it, it has increased its reserve food materials, and next what has happened, its nucleus has undergone division, and ultimately two cells, ultimately two unequal cells have been formed, which are the vegetative cell and generative cell. Now, the moment the microspore has undergone mitosis and it has produced two cells, no more we call it as microspore. It is now called as pollen grain. Pollen grain, or it is also called the male gametophyte. Please understand this concept. Usually people confuse with the microspore and pollen grain. Microspore is a single celled, single nucleated structure. But the moment it undergoes mitotic division to form two cells, it is no more called as microspore, but it has germinated, or it has divided to form a male gametophyte, right? How can you call it microspore now? It is no more spore. It is not a concept. It is not a part of sporophyte anymore. It is it has got its own identity. So you call it male gametophyte or pollen grain. Okay, so this vegetative cell is having lot of reserve food material and all, and vacuoles also, because it has to develop into pollen tube later. And this generative cell, it is spindle shape. If you observe, it is spindle shaped and it is suspended in the cytoplasm of generative cell. This also you have to remember. Generative cell is suspended in the cytoplasm of vegetative cell. Okay, next what happens? Okay, this is how many celled stage it is? Two celled stage. Two celled stage of pollen grain, and you should remember that in 60% of angiosperms, what happens? Anthers dehisce and pollens disperse. You have to understand the difference between dehiscence and dispersal. Dehiscence means anther is opening up. Okay, whereas dispersal is pollen grains are getting dispersed. Okay, so in 60% of angiosperms, anthers dehisce or the pollens disperse at two celled stage. Okay, then what other option is there for them? There is. If they are not dispersed at this stage, then what will happen? Or sometimes what happens? If they land on a stigma, if the pollens are dispersed and landed on stigma, right flower and right stigma, then they will undergo one more change. What is that? We'll see. This generative cell is there, no? Generative nucleus is there, right? That will again undergo a mitotic division. So how many daughter nuclei will be formed in mitotic division? Two. So what will happen? So if this is the vegetative cell, it will form, this generative cell will further undergo mitotic division to form 2 male nuclei. So this was 2-celled stage. Okay. Now here how many cells do you see? 1, 2, 3. So it is 3-celled stage. So in a 3-celled stage, which are the cells present? One is vegetative cell and other two are male gametes. One second. Other two are male gametes. Okay. So if in 60% of angiosperms the pollens get dispersed at 2-celled stage, in rest of the 40% of angiosperms, the pollens get dispersed at 3-celled stage. Okay. So this process of development of microspore and ultimately the production of 2 male gametes is known as microgametogenesis. So we will just quickly recall what exactly is happening inside an anther when the flower is just blooming or just planning to form. Okay. That time what will happen? Archisporial cell, which is a diploid structure, it will undergo periclinal division to form 2 layers, parietal layer and sporogenous layer. Parietal layer on the outer side and sporogenous layer on the inner side. Of course, parietal layer will form parietal layers of cells, whereas sporogenous layer will give rise to sporogenous layer will have sporogenous cell. Okay. That cell will undergo mitosis to form sporogenous tissue. Now in this cell, in this tissue, the cells will grow in size to form pollen mother cells, or they are also called as microspore mother cells. Till here all these structures were diploid. Now these pollen mother cells will undergo meiosis to form microspore tetrad, because one mother cell undergoes meiosis to form 4 daughter cells. Mother is diploid whereas daughters are haploid, and as they are the product of one single mother cell, pollen mother cell, they are clustered together like this. So they are called as microspore tetrads, but as anther mature and as dehydration happens, there will be a lot of water taken up by the cells. So there won't be much water left, that is why they segregate or separate to form microspore. So this process is called as microsporogenesis, whereas next what happens, microspores, individual microspore that will develop in size, it will store lot of materials in it, it will develop vacuoles, and ultimately the nucleus will undergo mitosis to form 2 unequal cells, which are vegetative cell or tube cell containing vegetative nucleus, and generative cell or generative nucleus, which is spindle shaped and it is suspended in the cytoplasm of vegetative cell. So the moment microspore becomes 2-celled, it is no more called as microspore, it is called as pollen grain, which is the male gametophyte. So in 60% of angiosperms, the anthers dehisce or pollens disperse at 2-celled stage. Okay, but if the pollens are dispersed in this stage and they have landed on the stigma, this is one option, or if they have not dispersed yet, if they are still in their flower, then what will happen? This generative cell, it will undergo further mitotic division to form male gametes, thus forming a 3-celled pollen grain, that is vegetative cell and 2 male gametes, altogether 3-celled structure. So in 40% of angiosperms, the pollens get dispersed at 3-celled stage. So pollens are dispersed, and if they land on the right stigma of, if not the same flower or if not the same plant, or if it is a different plant, then what will happen, this vegetative cell, it will develop something called as pollen tube. Okay, pollen tube, and of course how pollen grains are taken to different flowers, we know the beautiful agents, and that process is known as pollination, which is carried out by different organisms. We have to learn about pollination also. But yeah, if they have landed on the stigma, they will develop a tube-like structure which will pass from stigma through the style to the ovary. And that tube, that pollen tube is developed from vegetative cell, and this pollen tube will be carrying two male gametes, and that kind of fertilization or movement is known as siphonogamous. Siphono is straw like or tube like. So because tube is formed, it is called as siphonogamous kind of pollen pistil interaction. Okay, anyway, we are going to learn in depth about them in the further classes. So that is all for today. So development of male gametes from microspore, development of pollen grain which contains male gametes from the microspore is known as microgametogenesis. I hope this class is clear to you all. Thank you very much.